Welcome to this Frequently Asked Questions webinar. My name is Miguel Toribio-Mateas, and I am Director of Research and Innovation at Atlas Biomed. Today, as I mentioned, I am going to be sharing some of the questions that have been coming through to our customer services about the tests, how to take a sample, from very simple things like that, how the test samples are actually analyzed, to a bit more complicated questions. So I've chosen a few to start, and I wanted to let you know as well that we'll be running these sessions a bit more frequently. So if you have any questions about the test that you've been taking on yourself or sharing with your family and friends, please do share them with us, and we'll take a look at those in future sessions if we don't get the chance to answer them today. So first of all, the learning objective today is easy, simple, is to answer the most frequently asked questions that you send us on a regular basis. And I think probably we'll start with the most essential question of them all. How do you test my sample? Which is pretty straightforward, right? So first of all, you take a sample. You use the bottle that comes with the kit, and you take a small amount of fecal matter, and that is key as well, because you wouldn't believe the amount of questions we get about this. How much poo do you actually need for an Atlas test? And there's loads of different tests out there that use different methods. And the Atlas test uses what I like to call the Rolls Royce of the bottles for microbiome testing, which you're seeing right now on the screen. Most of the cost of the test actually is the bottle. It's quite an expensive bottle because it does a lot of the process that is involved in getting your report ready is pre-done by the liquids that are the reagents that are in that bottle. So that plays a really big part. So first of all, you register your kit. So you need to get your kit and register it online, and that you prepare the toilet with the cute little paper that sticks on the sides of the toilet seat like a post-it note, and that's going to pick up the poo when you sit at the toilet and unscrew the bottle and ready for collection. You pick up a little bit of fecal matter and basically just put it in the bottle, send it back to us in the box provided, and sit and wait for the results, basically. And in the meantime, what I wanted to say before I go into the next slide, very, very important, and the key to this question, the key answer to this question: how much poo do you need? So it's about a pea size. So the same amount of toothpaste that you would put on your toothbrush. So that is the amount, a tiny amount. So some people are used to other methods of testing or to other labs that perhaps use bigger bottles. They don't use the method that we use, which is called cysteine-X sequencing, and we're going to talk about that in a moment. So that is a very important distinction for the Atlas test. That is a tiny, tiny amount, and you need to put it in the bottle, give it a bit of a shake, and then just put it all in the sealed plastic container that comes with the box. The box is prepaid, so you stick it in the post box in the UK. So if you're watching this from anywhere else at the moment, we are, after Brexit, experiencing some issues with the posting to Europe and from Europe. So while we get those resolved, please, this only applies to the UK, and we'll get those sorted as soon as possible. So what do we do with that sample when it gets to our lab? So we get the bottle with your sample in there, and we do a number of things. So it's a long process, and that's why it takes a while for you to get the results. It's not something that we can just do overnight. we don't look at your poo under the microscope and, we know all the bacteria that are there immediately, and then we send you the report the following day. I wish it was that easy, but unfortunately it's not. So the first thing that we need to do is just to take out the mixed water or the liquid that is in that bottle with your poo that you have actually shaken. So it's been in transit for a few days until it gets to the lab. And in the lab, we extract the genetic material, and that's the genes of the bacteria, so to speak. And we focus particularly on a particular gene that is called, or a portion of a gene is called the 16S. And the reason we focus on that is that because it's a very characteristic piece of genetic material. So it's almost like identifying somebody by the profile of their nose. And if they have a nose that is very characteristic, or ears, or, eyes. So if you saw Prince Charles with the face completely blurred, but you saw the ears, they are quite characteristic, bless him. So if you saw, Robert De Niro's nose, that's quite characteristic, you might recognize him just even if you didn't see any of the other features in the face. This is the same kind of thing. We're trying to identify bacteria by making absolutely sure that we get the right feature identified, and that we do that through a method that is called PCR, or polymerase chain reaction, that is heating the genetic material, heating the sample at really high temperature for a split second, and that actually releases all of that information. And we do that a number of times to make sure that it's not just a fluke. So we need to be absolutely sure that when we give you the results, they are 99.9999 certain. So we do this, and there's a huge amount of data that is generated. So it needs to be put through a machine that is called the sequencing machine. And, it looks like a big photocopier kind of thing. And that consolidates all of the information that's come out of your sample. And that's called pooling the data. So it's just putting it all in one single place. And we also determine the basic structure of that genetic material, and that's called the sequencing. So again, we do that. We read that material a number of times, and again, if you're a bit more technical and you're used to reading this kind of literature, you will know that there is such a thing as reads. So how many reads? So it could be from 1,000 times to 100,000 times, so to speak. And according to how many times you confirm that the data is correct, then that's going to have more of a certainty. It's going to be more, it's going to give you more reassurance that what you have identified is actually the right thing. So that's a really important part of the process, because you want to know that when you get your results, they apply to you. And that's also a question that we get a lot, and I haven't actually put that in the presentation today. But I'm shocked that I have so little this, or I'm shocked that I have so much of that, because I never eat, whatever food that is associated with having so much of that bacteria, or I eat loads of this, yet I have very few bifidobacteria, which I should have a lot of them, because I eat a lot of a lot of pickles. It's never straightforward. Your gut is never going to be as straightforward as you eat something that gives you bifidobacteria, and then suddenly you get loads of bifidobacteria as a result 3 months later. I wish it was that easy, but unfortunately it's not. And you need to think about the gut as almost like a person in the real world. You can have it all planned out as you want your life to be, and yet life throws you so many curveballs that you cannot anticipate what's going to happen. And your gut is a little bit like that. It's a living organism. So if you look at it that way, maybe it makes a little bit more sense, because we try to control it too much, and it's not about controlling it. But let's see why this is important. Well, it's important because the 16S way of working is considered to be the gold standard in microbiology. It's supposed to be the gold standard in microbiome sequencing. And again, that sequencing is what happens in those 3 last steps. So the location of that portion of genetic material, the 16S, that is the characteristic, the feature that we think that is Lactobacillus longum, or that is Bifidobacterium brevis, or whatever it is. And we know certainly that that is the case because we have read it a number of times and we've confirmed it. So that's really important because, again, it gives us the reassurance that that is what scientists around the world are confirming, that is the gold standard way of identifying bacteria. And that leads me to many other questions, but I'm going to just pick up one, which is, for example, can you identify things like parasites or candida? And we're going to talk about that in a second. But this way of working, this way of breaking down the features, the characteristics of your microbiome, to be absolutely certain that what we are giving you is correct, is again, we could say that is 99.9% accurate at the time that we give you that sample. And again, you might be shocked in a positive way or in a negative way. The good news is that the gut is always in flux, is always changing, and whatever happens, whether it's negative or positive, you can always kind of spin it around a little bit. So that is what allows us to break it down into the areas that you see, from the more general to the more specific that we have provided you with in your report. So that's a really important thing to take into account. But then again, it's important because fungi, for example, are a lot more difficult to identify. They lack that essential feature, that, those characteristic ears or nose or hairline or hair color that you could say, oh, that I recognize that person immediately when you see them on the telly. If you transpose that to microbiology and trying to identify a microbe, fungi are a lot more difficult to identify via this method. So there are other methods that are actually very appropriate for the identification of fungi, and microscopy is one of them. So just looking with advanced microscope techniques at a fungus like candida, for example, can be a really good way to identifying it, but it brings some certain margin of error that is actually higher than the 16S sequencing way of working. And also it requires that somebody who is very specialist in the identification of microbes, whereas we're relying on a machine that is 99.99999 correct. I'm not saying that people cannot be that correct as well, but you're relying on the laboratory person to say, okay, this is candida. What type of candida? Is it candida albicans versus candida something else? So that you're relying on that kind of human component. So when we get the question, can you test identify candida? The answer is, at the moment, no, but everything changes in science so rapidly that there has now been a way to utilize the way of working with bacteria, with other microorganisms, such as candida, for example, or such as parasites like blastocystis, for example, which is, again, something that pops up in the human gut a lot of times, or the Diantoamoeba that, again, pops up in the human gut very frequently. And in exploring those new methods, we are probably very soon to release a test that will accompany the fungi and potentially other microbes as well in the gut. So that is something that I wanted to say because it keeps coming back to us a lot. And also in terms of the pricing, that the influence on the pricing of the test, that again, it would make it potentially more expensive as well. And that's one of the things that when you are a commercial entity, a company, dealing with you people there in the real world, and we want to give you a accessible science about your gut. If you start piling up loads of add-ons, it ends up costing a lot more, and not everybody wants the fungi, and not everybody needs to know about the fungi. So the fungi are tricky, it's just like bacteria, they change all the time. And I've seen it too many times that people get a little bit of candida in the gut, which could be absolutely normal. They go on a rampage of anti-candida dieting or cutting out loads of things that they enjoy eating, and they basically upset the gut ecosystem as a result. So that's something to take into account as well, that it's not always about killing something, it's more about the overall picture. And that's another really good example. So we get loads of questions around, how do I get rid of whatever it is? And this is just one example. So the Desulfovibrio is a type of gram-negative bacteria that being gram-negative actually carries LPS. So we talked about that in the previous webinar, which if you haven't watched, you can just go on the Atlas YouTube channel and watch it. So I explore that as part of the impact that it has on immunity, and the fact is that LPS producers, and the Desulfovibrio is one of them, being a gram-negative bacterium, carry this thing called lipopolysaccharide, or LPS, in their membrane, and in carrying that in the membrane, then what they do is, anytime that they touch any surface in the gut, including the gut lining, they're going to elicit a response by that gut lining that is completely loaded by molecules involved in immunity. So it's probing your immune system, basically, that LPS, and it acts as a toxin, as an internal toxin or endotoxin. So it's a tricky kind of thing. But again, just like anything in life, like going back 20 years, there was no microbiome talk, but there was oxidants and free radicals and antioxidants, and everybody was jumping on the bandwagon of more antioxidants will keep us wonderful, young forever, and we need to quench all the free radicals, whereas the free radicals are actually a very interesting kind of phenomenon, because they are communication molecules. And now we think that LPS is actually very toxic, but why is it there? Why is it carried by some microbes that are actually living in the gut? So that's another interesting question. And perhaps in 5 years' time, we'll think about that in a completely different way. So that's something to take into account as well. But in any event, gram-negative bacteria do carry this LPS. Again, it plays a role in immunity, in regulating immunity, in upregulating immunity, because it basically makes your immune system think that there is an attack from the inside when there is more LPS. And obviously the Desulfovibrio, if it carries along with many other bacteria, so I showed you just a few in there. So the LPS producers, if you have any of those bacteria, I'm just going to show you a few in a second, then you'll have more chances to be probing the immune system on a regular basis. But and of course there's consequences as well, like persisting inflammation and so on. But that's not the only thing that will be making you feel like you do. So it's easy to just focus on one thing. And if you're feeling unwell, or if you have bloating continuously, it's easy to just want to blame one thing, because it's easy to just resolve that one thing and then that goes away. But it's often about loads of tiny little steps, as opposed to just one big step against something in particular. So it's just changing that kind of way that we look at things slightly. And I'm particularly keen to explore the fact that the Desulfovibrio and the family that it belongs to, and the whole type of bacteria that it belongs to, which is proteobacteria. If you zoom out and you look at the type of microbe that it is, proteobacteria are slightly different to other types of bacteria, because they are in general more linked to inflammation. So you've got that kind of a balance between, again, inflammation and anti-inflammation front on the gut. on the gut lining, the effect that certain types of bacteria are going to have on that gut lining. But that changes over time. So if you have a person, this is a real example, anonymized example of somebody who's taken the test a number of times, and you see that a family going up and down, and in response to different things that are happening to them. So diet obviously is really important, and at Atlas I'm passionate about making changes to diet that are conducive to better health. But at the same time, we cannot reject the fact that there's a number of different things going on in our lives. So there's stress, there's pharmaceuticals, and not only the antibiotics, but I mentioned ibuprofen last week, so that has a huge impact on actually decreasing the amount of stomach acid, because it inhibits both COX-1 and COX-2, which are enzymes that are needed for inflammation. And this is why ibuprofen is wonderful, because it takes away that inflammatory response. So it takes away the pain that is generated by the inflammation. So if you have a headache and it's an inflammatory headache, it goes away, or period pain, or arthritis, or whatever it is, it goes away temporarily. But at the same time, you're making your stomach work in a deranged way. You're saying to the stomach, don't bother producing so much stomach acid, because you're fine. You're sending up kind of like a perverse kind of message to the stomach: do not produce stomach acid, because those enzymes are involved in many things. Some of the things that they do is to increase inflammation, but also to produce stomach acid. This is the body. It's complex. Not one single element of it. That's just the one single thing. So it's taken into account things like where you live, whether you live in a farm or you live in the middle of London or Manchester. That's going to have an impact. And again, the gut-brain axis, that connection between the gut and the brain, is really, really powerful as well. And I couldn't say it anymore. I say it every time that I have a session. So of course, sulfate reducing bacteria can be tricky, and the Desulfovibrio is one of those. It gives your body access to sulfur, and in doing that, there could be bloating. There could be bacteria that actually travel to the small intestine, or try to go up into the small intestine, making you feel bloated quite high up in the colon. That feels quite uncomfortable. There could be obviously gas that smells not in a very nice way. So all of those things, I'm only too conscious they are not pleasant to experience, but trying to kill just one thing is often not the way, because even if you follow somebody's advice who tells you if you take garlic, that will go away. Garlic might actually reduce that, but it may also feed other bacteria that also give you bloating. So in the end, you don't know that you've actually experienced the benefit, because the bacteria you're trying to kill has actually been killed or reduced, so to speak. But at the same time, you've allowed other bacteria to grow that are also making you feel bloated as a result. So it's a fine balance, and that's what I wanted to illustrate in there. So we do have a new section in the report that is available from the dashboard. So if you go to the dashboard and you find inflammation and the microbiome, which is, you can see where the blue arrow is, and you click on that, then you get your inflammation report that goes from 0 to 10. And that tells you about the kind of bacteria that contribute to inflammation by a number of different processes. But because things are complicated, we've figured out that identifying bacteria that produce LPS is one of the best ways to give you this kind of ratio between the inflammatory or pro-inflammatory bacteria and the anti-inflammatory bacteria. So we calculate that for you, and that gives you an understanding. So if you retest, which I can never encourage enough, doing one test is interesting, but then it's like, life goes on. And as you change and you do different things, then that can be a very interesting way of finding out what the effects of that have been. So retest and actually check what is going on with your inflammation and your anti-inflammatory potential as a concept to grasp and try and explore. So another question, a very interesting one as well. What does it mean to have high Oxalobacter when I don't eat loads of oxalates? And this is quite specific, but also I can extrapolate it to a number of different situations that are exactly the same. So why do I have really high, and again, it's never just only the bugs that we think about as being bad as well. It could be high Akkermansia. Why do I have 10% of Akkermansia? Should I be worried because it's in the green? There's no bad level known for Akkermansia, but we do know that it should probably be just around, 0.5% of your microbiome, if that. So if you have 10%, what does that mean? Well, it means, if you have a bottle and 10% of the bottle is Akkermansia, there's going to be only 90% left for all of the other things that should be in there, hundreds of different things. So if it should only be 0.5%, no doubt there's going to be an impact, whether it's positive or negative. So there's that kind of thing. But particularly on this one, so oxalates come from a range of sources. They come from things like spinach, for example. So you could think, again, in a very mechanistic way, in a very linear way. I always think of this way of thinking like a sausage machine. Whatever you put at one end of the sausage machine comes out the other end. So if you happen to put tofu, because you're making vegetarian sausages, tofu comes out in maybe a spiralized, so it comes into your sausage casing, obviously, but it is tofu at the end of the day. It's nothing, you haven't transformed it, you've just basically channeled it into the exit point. When we put something in the body makes such a complex set of changes to that food that it ends up being nothing like what you've eaten. A typical example, I've got a sick dog at the moment, and he's very anemic, and I keep thinking, even though I'm used to this complexity thinking, I keep thinking, why not give him iron? And the vet tells me, but he's got a problem with the way that he holds on to iron. So giving him iron is not going to do anything, because you could give him iron every day at high dose, he's just going to pee it out, because the way that the iron should fix to his cells is not going to work. So this is a perfect example. And with oxalates, it's just the same kind of thing. You could eat spinach every day, and you could struggle to have a high oxalate thriving bacteria, on the basis that they might be affected by other bacteria that also like whatever the spinach is giving you. And because the spinach is green and it's got some nice kind of like fibrous structure to it, it could be that it's also feeding some Faecalibacterium, it's feeding some Rosaburia, which are fabulous bacteria to have in your gut. They are really anti-inflammatory, they produce butyrate. So in feeding them, you're reducing the ability of the Oxalobacter, which may be a little bit more delicate, it may be a bit shy, some of us don't mingle in social situations. Think of the gut like that as well. So you might have a colony of bacteria that are all outgoing, and they are the soul of the party. They come into a room and they take over. You could have some of the ones that they need a little bit of encouragement, and they don't really like talking to people very much, but they do have the heart in the right place. They do their things in a right way. They provide you with benefits. So try and think about it that way, and also think about the fact that your microbes are constantly changing from birth until you die. There's a change in diversity, and in diversity changing, there's going to be a distribution in those different bacteria that you are actually carrying with you wherever you go. So that's a really important matter to take into account. So that probably answers that question. I also want to answer another one that we get all the time. And when I've been working in clinical practice myself, I don't do so much now. I used to get this all the time. People are desperate because they are bloated or they are reacting to different foods in an unexpected kind of way. And there's never an easy fix again, but my advice is to try and take things little by little, if you can, as opposed to go full blast. And of course, if you pay a professional to give you advice, they're going to give you a lot of different tools to play with and to work with and to try and experiment with. And that is exciting. At the same time, if you're introducing too many different aspects to play with at the same time, you have more chance to not know which ones are actually working and which ones are actually upsetting you. And that is a trick, kind of it's a tricky situation, because obviously you want to feel good, so you want to put as much effort into changing as possible. But I would say start with a diet diary and try and break down what you do, what you eat for breakfast, lunch, and dinner, and your snacks and drinks and everything else, everything you put in your mouth, literally, including supplements, everything. Take that down for a week and see if you have any patterns in there that you can actually identify, because that can be a really useful way to figure out whether there's something that you keep repeating every day and you hadn't realized it's just because it's a food that you're really drawn to, and you end up eating, I don't know, tomatoes every day. So could it be the tomatoes? So give yourself a chance to actually not change anything for a week and just observe, like a field kind of field work. You're observing in a archaeology kind of situation, but you're observing yourself. You're doing a bit of ethnography to yourself. So do that, explore what is going on, make a note, and then the following week, just sit there with a cup of your favorite tea or a glass of water and just go through it and try and pick up the themes. Is there is tomato there every day? Is bananas that could be, are you having the same breakfast every day? That needs changing. What is it? And of course, there's that some easy ones. Some people say to me, oh, it's always the gluten or it's always the dairy. It's not for everybody, but it could be. But, if there is gluten and dairy at every meal, are you having bread or a variation of gluten-containing grains at every meal? And how could that be affecting you, especially if they are more processed than other types of grains that may be less processed? Like, for example, sourdough bread should only have flour, salt, and water as the ingredients. And if you pick up a loaf of your favorite brand bread in the supermarket, I can guarantee you it will have probably 20 ingredients. So that kind of thing. And again, I am aware that there's a range of people watching this, and you will be one of the savvy ones who thinks this is like 101, Miguel 101. There would be somebody there who's never heard about this before. So I need to be kind of, right in the middle. Because if we get into the very, very complex situations, then it can be quite niche. But I appreciate this. I need to be a little bit general. But also it's about when you eat. And I praise my friend and colleague Janet Hyde, who wrote these books back in 2015, 2016 actually, and the Gut Makeover, and that was an Amazon bestseller. And the recipe book is a wonderful book as well, and it's science-based. At the time, it was for me the best book that there was on the gut, before everybody jumped on the bandwagon of the gut as well. So I think he was a trailblazer of a situation with Jeanette and writing these books. And she kept saying about wanting to write a book about fasting and or time restricted feeding. And there's a lot of science now that talks about the value of not just looking at what you eat, but when you eat it as well. And if you are somebody whose gastrointestinal system is overloaded and it's shouting at you, give me a break, and it will be shouting at you, telling you, give me a break, when you're bloated continuously, when you're cramping, when there is, of course, it may be shouting at you, but for other reasons as well. So, of course, talk to a doctor if there is blood in your stool, if there is excessive mucus in your stool, if you feel pain, because those are red flags. And we're talking about non-clinical situations or subclinical situations that are difficult to kind of ascertain what they are. If you end up having Crohn's or ulcerative colitis, there's no point in just like fiddling around with a few lentils. You need to get seen and get to the bottom of it, and then figure it out with the diet as well on top of that. But you need to know what it is that's going on in your gut. But if you haven't got a clinical situation that you're dealing with, or you've got this kind of a undiagnosed IBS that many people do have, including me, I cannot recommend enough just having a look at giving your gut a break. And I think just eating for 10 hours as opposed to just thinking of fasting, which people still get a little bit freaked out about. I don't know what it is about the word fasting, but I think some people just don't like it. They just think they're going to faint or something, which is never going to happen. But, it's just allowing yourself to eat during a window of time that could be 10 hours, which is completely doable for most people. If you have a semi-early dinner and a breakfast that is a little bit later, or you skip breakfast altogether if you are that way inclined, that can give you a nice kind of a break from the food. That if the first thing that you put in your body in the morning is also some kind of therapeutic thing. It could be, if you're that way inclined, something soothing like an aloe vera shot, for example, or it could be some soothing tea like linden tea, for example, which is not only soothing for the gut because it's really rich in mucilage, which is this kind of like mucusy. If you've drunk linden tea before, linden flower tea before, it's soothing for the nerves. In countries around Europe, it's used. Spain is massive. It's tea for the nerves, basically, to calm down, to have a good night's sleep, and to calm down the nerves when people have anxiety and so on. Massive in Germany as well, which has a really big history of natural medicine as well, and it works on helping you release more GABA as well. So it calms down the brain. So it works on that kind of a gut-brain connection as well. It's a lovely, nourishing tea. If you drink it in the morning, it's not going to make you fall asleep, but it will give your bugs some kind of mucilage, which is nice for anti-inflammatory bugs like the Akkermansia, for example. And you also have some nice polyphenols coming in from there, some flavonols that are interesting for the body. If you cannot be bothered with linden tea, or it sounds too exotic, what about your green tea? Your green tea is a fantastic source of all sorts of catechins, which are, again, kind of like polyphenols, incredibly antioxidant, but also nourishing for the gut, and reducing the free radical activity that goes on when you have things like the Desulfovibrio, or you have an increased Klebsiella, or whatever has come up in your report and you're worried about. Or if you have a report from another lab and you think, you didn't talk about whatever, that was in my H. pylori, for example, which is another tricky one to identify as well. And we keep trying to find out whether we can apply the method that we use to identify it reliably. Just choose something nourishing that first meal in the morning, and that's going to give you a lot of opportunity to work with your body and to allow to give your gut that break that is requesting from you. So these are the questions that I wanted to cover today. Unfortunately, we ran out of time now. I hope that you enjoyed the session, and I hope to see you very, very soon at our next webinar. Until then, take care of yourselves, stay happy, healthy, and see you very, very soon. All the best. Goodbye.